Okay, we are in section 3 of your student notes for chapter 5, section 6 and 8 on transport. Active transport is when materials move up or against the concentration gradient from an area of lower concentration to higher concentration. This requires energy from the cell and thus is called active transport. I have a picture on the right of a sodium and potassium pump, which moves sodium ions and potassium ions against the concentration gradient. It says note energy from ATP is required, and it cites your book. But I have a picture here. You've got your cell membrane. ATP is going to come in and donate a phosphate to this protein. The phosphate will bond to the protein, and three sodiums, a third sodium comes in, and 3 sodiums will be exported out. ADP is coming off of here then. And once the sodiums leave, then 2 potassiums will come in. The phosphate gets kicked out of here, and then the 2 phosphates will come in. So if you notice, it changes shape as it goes across, and you are pumping against the concentration gradient. There's more potassium inside than there is outside the cell. So let me walk you through that again with your notes. 3 sodium ions are released to the outside of the cell, and 2 potassium ions are brought into the cell. There's another type of active transport called endocytosis. Endo means in, cyto means cell, osis is condition of, so you're moving things into the cell. It is the movement of large molecules or groups of molecules into cells. The cell membrane will fold around these particles and engulf them. Phagocytosis or phagocytosis is a type of endocytosis which engulfs bacteria, and I want you to draw a pretty diagram. So I have a couple of diagrams of this happening. Here is a picture of a bacterium. It is being caught by receptors, so the cell knows to engulf around it. Phagocytosis occurs and it pulls that inside, it wraps the cell membrane around it and forms a phagosome or a phagosome. The lysosome, with all these enzymes, will merge with the phagosome and make phagolysosomes. Once it gets broken down, then it will go through exocytosis and expel the material out of the cell. I have another picture for you. I don't think there's another one on this page, but I did want to show you again. This is another example of endocytosis. Don't hate me, I did use Wikipedia. There's a kind of a schematic up front. That's not where I wanted to go. It lost my picture. I'm so sorry, guys. But if you just Google it, there are plenty of pictures. Oh, here we go, of endo and exocytosis. So here's endocytosis. Again, a solid particle like a bacterium will come in. The cell membrane wraps around it and engulfs it, and it brings it in. It might happen in pinocytosis, which are small dissolved particles, and it makes a vesicle and brings them into the cytoplasm, or receptor-mediated endocytosis, where it bonds with receptors and then it brings those into the cell. It looks like it did it again to me. Here's an example of phagocytosis and exocytosis. Exocytosis is your next example. It's basically when a vesicle will merge with your cell membrane and then expel the material out of the cell. There's another good picture in this section, but I think my search, for whatever reason, is not helping me this time. This one is the one I wanted. Maybe it's here. Oh, here's one. The cell membrane the vesicle will come to the cell membrane, it will merge with the cell membrane, and it will expel it out of the cell. You could draw that one. Here's another example from a side view where the Golgi apparatus is merging with the cell membrane and expelling materials through a secretory vessel. Here's another one where it's coming out and merging and separating it. A cool fact, and I liked this one. This is the one I was trying to find because it's got the phospholipid bilayer, and it shows how that's even part of a vesicle, and then how that vesicle will merge and expel the material outside of the cell. One of the places that your body uses this is in nerve cells. Oops. Nope. It should have been on that other page. But your body uses this process in nerve cells to send signals through your cell. It happens constantly. It happens in the pancreas, to expel hormones and different things. Here's an example of it happening in neurons. It will go through exo and endocytosis. Well, not endocytosis, but it goes through exocytosis to expel those, and then those will attach to receptors and cause things to happen on the inside of the cell. There was a picture of it happening on a neuron somewhere that I've misplaced. It's not showing up anymore. But there's some diagrams. I hope that's helpful, and that's your notes for Chapter 5.